Oh, yeah, all right. Well, so how do you, is there a way to also go back? Yeah, okay. All right, so well, we talked a lot about the hallmarks in here, drug resistance and so forth. I don't think I need to remind you of the different types in which resistance can arise, so, and why it's such a major resistance to therapy, that is to say, why it's such a major problem in cancer recurrence. But let me remind you a little bit, and this is more of a philosophical discussion, and it'd be good to have over coffee break or whatever, much more discussion about this, the kind of issues I'm going to bring up. So the sort of paradigm that we're used to is a selection paradigm. Namely, we assume that the random changes that can occur, whether genetic or epigenetic, in a cell line, these random changes will be such that maybe we have some pre-existing heterogeneity. The application of a drug changes the fitness, changes the environment, and hence the relative fitness of the different populations. Typically, a resistant population might be growing less fast than a non-resistant population because it needs to express things like MDR or other mechanisms of resistance, okay? However, once we are we 30 minutes. 30 only? Okay, all right. So, so I have to say, and I have 64 slides. Okay, that's okay. At the end, I'm going to speak faster. You got scared with that, yeah. I actually once gave a talk in which the number was lower, but then when I got to the end, I had negative numbers. So the audience was anyway. Let's see, now I just wasted 15 seconds. Anyway, you change the fitness and because you change the fitness landscape, and because of that, the guys that were sensitive now, of course, will die when you apply the drug, and the others will take over. Okay. Now, what I would like to, and typically we go back to Lurie and Delbruck in the 1940s in the context of bacterial experiments, this whole theory that was developed to understand the difference between pre-existing variation and variation that arises during treatment, right? So this is a classical area many people have studied in the context of cancer as well. But I would like to focus on a subtler question, which is very difficult to explain to biologists, I find, because the answer typically is, so who cares? It's the same thing, and I want to try to explain to you why it might make a difference. So the question is, so we have pre-existing variation, right, and Darwinian selection. We also have the idea of random changes that may occur during treatment and Darwinian selection, but we also might have what one might call Lamarckian changes, and Lamarckianism is making a comeback, right, in a certain way, right, where we think of the fact that the application of the drug, think of it being in a different environment, actually causes generation of variation. In other words, the paradigm is not the classical Darwinian paradigm of random changes and selection, but now it's a paradigm of induced changes, generation of variation. And by the way, Darwin, if you go back and read, he was aware of that. He was an anti-Lamarckian. I mean, he was talking about this little, what do you call this? Little molecules, well, he had another word, molecules that could go into your reproductive cells. And, so he sort of was much smarter than people think, even anyway. So why am I interested in this? We started with the work that we're talking a lot with a particular group that did this work, in particular, Angelique Stephanou and Shui Wang at the Institute for Assistance Biology in Washington, in Seattle. And they had this very interesting work. There's other people that have done sort of similar things, but their paper was particularly interesting, in which they were exposing a particular kind of cell line. It happened to be some leukemia cell line, but it's not important for the purposes of the concepts. They were being treated with a particular chemotherapy agent, and this could apply also to targeted agents and so forth. It's just generically, but in the particular experiments with this particular agent, and in the wild type, before you apply the chemotherapy, the cells are randomly distributed according to some expression, in this case of MDR1, multi-drug resistance 1, which is a gene that expresses a protein that makes some pumps that takes things out of the cell, okay? And you have some sort of a histogram of expression of FDR1 in the wild-type population. Now, when you apply the drug, it turns out that the cells tend that obviously the sensitive cells get killed, you end up only with the resistant cells. Okay? Now, what they found is if you take these resistant cells and you stop treatment, you reconstitute the actual wild type probability distribution. So there's some sort of stochastic process, and you have some sort of equilibrium state of this stochastic process. Okay, you've moved it, you force things away when you apply the therapy, but then when you take out the therapy, the thing goes back to the previous equilibrium. Okay, so the question is, clearly there you have that the change is being caused by the drug, because you took out the drug and you go back to the previous state. Okay, we heard many people talking about these kinds of things during this week. They also found, they did single cell tracking to see this. It wasn't just a population question. It's a beautiful paper, lots of really cool data. They found that the effect is dose dependent, and a number of other things that, I'm going to talk about. So, and by the way, they got a repopulation after a couple of weeks of the, meaning going back to the wild type histogram. There are a number of different papers. This is a random choice of papers. Again, the slides are a little old by now, but there's much more. So what I want to emphasize is that if we're talking, so the usual paradigm that we know from Lurie and Delbruck and so forth is pre-existing versus appearing during treatment. And what I want to say is the appearing during treatment has two subtypes, whether it's randomly happening and the appearing during treatment is because there's Darwinian selection for those, and the other sort of subtype, which is we generate variation and then we select. And you might ask, as I say, when I talk to biologists, it's like, well, who cares? At the end, it's the same thing. You get resistance or whatever. But what I want to emphasize is that when you look at the problem mathematically, you can actually differentiate between the two, even with the simplest, most stupid mathematical model you could think of. And that has consequences, or at least potential consequences for therapy. And this is what I want to explain. All right. We had papers. Actually, this paper appeared already a long time ago. Sorry, the slides are very old, okay? And we have a new paper that we're working on that I hope to present, like I said, 3 weeks from now. Those talks are going to be much shorter, actually. All right. So what is the model? The model couldn't be simpler, okay? Is the absolute baby model. We started with more complicated models, kept simplifying, and really there's almost nothing. It's the simplest thing. We have two populations, sensitive cells and resistant cells, okay? Generically resistant cells. We have growth rates for each of them. I'm putting a logistic term in the, now we're doing some data fitting. It turns out that we don't need the current capacity is so large for the in vitro experiments that it doesn't matter. But we have two growth rates. We're going to assume that the growth rate of the resistant population is less than the growth rate of the wild type population. We have a rate due to the drug, which I'm taking just a simple mass action kinetics kind of thing, proportional to the U of t is the amount of drug at time t. Okay, there are t, so this is S of t, of course, but I put the t explicitly there because I do want to talk about time varying protocols. So there's some sort of constant there that tells you how much the killing is effective. And I'm going to assume that the killing is more effective on the sensitive cells than on the resistant cells. So there's no cheating, is the obvious thing. I'm going to have a term, negative epsilon s plus epsilon s, which is a flux from sensitive cells to resistant cells that is independent of the treatment. So I'm assuming that there's some sort of exchanges at the gamma, which is a flux back. So I'm going to assume there's some sort of back and forth between the two is independent of the drug. But then in addition, I'm going to have a term that depends on the concentration of the drug with this parameter alpha. When alpha is equal to 0, that means there's no induced resistance. The resistance is that's purely random. When alpha is bigger than 0, that quantifies how much of the resistance is actually due and how is it dose dependent, right? How it depends on the drug. Okay. I think I haven't said more than that. We have competitive inhibition due to the carrying capacity, okay? V is the total volume with the sum of S plus R. So there's a competition between the two, and of course, what's going to happen is going to be quite, even if you didn't have alpha, you are going to have some competition between, even without inputs, you're going to have some competition between the resistant and the sensitive cells, right? Okay. So what I'm going to do first, and the mathematical analysis is simpler for that, you do some rescaling, some non-dimensionalization and whatnot, you end up with this parameter PR, and I'm going to assume the most important thing is I want to assume gamma is 0, so I'm going to assume there's no back mutation, so to speak, or back phenotype transition from R to S, just for simplicity, okay? And I'm also going to assume the resistant guys are completely resistant. It just makes it a little easier to do mathematical analysis. And then we do simulations, what happens when that's not true. All right, so first of all, it's completely obvious. I mean, you can write it as a theorem, but it's completely clear that no matter what you do, and I also normalize S plus R is equal to 1 at the beginning. Sorry, it's less or equal than 1. So 1 is the maximum magnitude. So it's easy to see that no matter what you're going to do, you're always going to converge to everybody's fully resistant. And because I'm not killing resistant cells, right? So no matter what I'm going to use, what schedule of treatment I'm going to use, I'm always going to converge to no sensitive guys and resistant guys. That's just the way the model is. And you could say, well, then what's the interesting problem there, right? So there is an interesting problem you can still set up, and it's as follows. This is just reminding you of the equation. So we're going to make the following, we're going to study the following mathematical problem. We're going to say that when there's going to be two threshold values of the total volume, V for V detection, and VC for V whatever, critical or whatever, I forget if I called it something else, critical size, yeah. So the idea would be we would like to get the total volume of the cancer. So as I said, eventually you're going to go, everything is going to be resistant, the volume is going to be maximized. But what I'm going to assume is that remember the carrying capacity that I had, right? Which I took to be K equals 1. That's why, okay? So what I'm going to ask as an objective is I would like to delay as much as possible the time I get to the critical volume. Okay, again, it's going to happen no matter what. So what we're going to do is how much can I delay things? So presumably, it's an idealized problem, but you can say at that point, you might have to try a second line of treatment or something, okay? All right, so that's really a mathematical problem. We want to find a U of T, given, the initial conditions, we could take to, for example, be 1, 0, all sensitive and no resistance. We like to find a way that when we start treatment, namely with the total volume go to this detectable state, we apply treatment, we know that eventually things are going to fail and they're going to go up again, they're going up to 1, okay? But the moment we want to delay as much as possible the moment that we cross the critical threshold. Okay, so that's going to be the mathematical objective. Once again, we can cure the cancer, what we can do is delay as much as possible in this trivial model, okay? Then you can make it more interesting, but just in this trivial model. Okay, so the first question that we could ask is, okay, so that's my gain. Simple question. Should we apply a constant therapy or should we apply some sort of pulsed therapy? I don't like the word metronomic exactly, which was suggested the other day. In this context, metronomic is fine. We have actually another paper, metronomic therapy, completely different, but I like to call it adaptive therapy. So somehow I want to be measuring what's happening and that I want to be able to it or not. And I'm going to make it even simpler. Rather than solving the general optimal control problem, I'm just going to ask the following question. Suppose that we can switch between one value of the drug and 0, and we need to find out at which points do we switch on and off. And to make the problem fair, I'm going to assume that the area under the curve is the same. So the total amount of drug is the same. Okay, so again, the mathematical question is, I'm given a budget on how much drug I can apply over, whatever, 30 days, 60 days, whatever. It doesn't matter. It's a mathematical problem. I can make any units I want. But I'm going to decide on how much drug I'm allowed to apply. And I'm going to compare applying it at a constant low value or applying it in a bang-bang way. Okay, as you would say in control theory. Okay, all right. So, and we took a particular case, and then the theoretical question, there are parameters and all this, so this is nonsense in some sense, right? Unless you have a real model, right? But we wanted to study the following conceptual question. Suppose that suppose that resistance that appears during treatment is only, and by the way, this is non-pre-existing because I'm starting at 1, 0, okay? So suppose that the resistance during therapy is all due to purely random transitions and there's no induction of resistance, which is this new idea that I'm trying to push for, okay? Well, not new, but, in this context. So that would be alpha equals 0. And let me compare that with what happens when alpha is non-zero. Why did I pick 10 to the minus 2? Just because, in the computer simulations, it was easier to work with that. It's nothing magical about that. Okay. So the question is, which is better? Which delays the most the therapy? And the bottom line is that, in fact, there's a big difference. If alpha is equal to 0, okay, where do I have it? I have to remember what's, yeah, there should be an, I don't know what it was. Yeah, if alpha spontaneous, right? If alpha is equal to 0, which I call the spontaneous case, there's no induction from the therapy, then in fact the best thing to do is to use a constant treatment. And if alpha is not 0, the best thing to do is to use this bang pulsed type of control. And in fact, it's actually it ends up being quite uniform in the solution. Okay, all right.
But
so
very close, right?
Yeah, well, that's because it's the optimal. So, okay, so this is an excellent question. So the way we stated the problem, we said there's a critical value, 0.9 of the maximum tumor size in here. There's a critical value at which the doctor will say, wow, this, it's everything failed. We need to go second line of drugs or something, right? So the optimal controller, purely mathematically, is smart enough to say, oops, I'm getting there. Let's apply another pulse. Let's go down. Then, of course, because of resistance and everything, it starts going up again. Whoops, let's go down. It's not exact, actually. It's very interesting. It's difficult to understand why it picked this, okay? Until, we are out of luck. There's no more, no more that we can do. And the constant one stopped, got there much earlier than the pulsed one, okay? In the case of alpha, this particular alpha, you can do different alpha, the graphs will look different, okay? Similarly, in the case of a purely spontaneous emergence of resistance, the pulsed one, the best pulsed one, which was this one, doesn't last very long. We get in trouble. Well, with a constant one, we go further. So, do I think that these graphs mean anything? Of course, it's BS. I mean, it's math. It's just, parameters don't mean anything, right? But what's important is a message. Okay, that at least in this formalism, at least in this way of thinking of things, we really say that therapy is better if there is induced resistance. Okay, if there's Lamarckian, let's say, versus Darwinian. Right, so I see this as a way of saying, hey, we need to think about these things much more, look at more realistic models, ask these questions in more realistic models. Okay, but at least from a mathematical point of view, in a very simplified model, we're getting this kinds of suggestion. Okay. There's a lot of, I don't know how much you're interested in optimal control theory. It turns out that this is not a classical optimal control problem. I wrote a textbook and I talked about optimal control and whatnot. And when I started looking at this, I thought it was going to be trivial application of things. And it turns out it's not. It turns out that this way you want to maximize as opposed to minimize. You can maximize a return, okay? You maximize a profit. But we're talking about maximizing time, this minimum time optimal control that usually leads to bang bang and so on. But to maximize the time of the viability of a set, if you know about viability theory and differential inclusions, and so forth, we're talking about there's a set that is viable. We want to maximize the time that we are inside this set before we exit the set. And that turns out apparently not to be a classical control problem at all. We had to develop a number of things. And that was that second paper that has a lot of the math. So, this is just saying what we said, okay? The optimization objective, we want to maximize the time that we stay in our set. In this case, S plus R less or equal than V sub C, which is this triangular area in here. We want to find the thing that maximizes the exit time TC. Okay. So yeah, again, is anybody interested here in optimal control? Okay, so We actually prove we use the maximum principle. We use selection under necessary conditions, sufficient conditions, forth. So necessary. Wait, I have it at the end. I'm forgetting by now. Anyway, 9 years, many years since we did this. And what we actually show is that the optimal solution has a funny structure. There is a switching between maximum and minimum dose, maximum dose and that's typical bang bang in optimal control. But then we have this funny what we call sliding trajectories, which are trajectories that actually make us slide. Did I have it? Where was it? Oh no, maybe actually it was here. Sorry. Maybe it was, sorry. Yeah, I didn't, it's not in this picture right now, it's in later, but basically things that make us sort of slide around here as long as we can before we exit and we lose the game. Okay, so it's a really funny kind of optimal control situation, right upon triaging maximum principle and everything, you know, the usual thing, but it actually gets complicated. This is just a numerical example of how things work. A little bit additional thing, we're interested in identifiability here. So good. So question, can you identify this parameter alpha? Okay, suppose I'm allowed, suppose I have a real, real system, I do a model, maybe it's larger dimension or whatever, but at least in this case, can we in principle find alpha from the measurements V of t? All right, is a parameter identifiable? The answer is yes, the parameter is actually identifiable. And you could use, actually you can identify it using a dose of 0, 1, 2, or a linearly increasing dose. If you try for those types of 4 types of inputs, you observe the volume V of t. From those, you can, in theory, identify alpha. Now, of course, in practice, there'll be noise. There's question of practical identifiability and all these things that we know. You might want to use profile likelihoods and so forth. But at least from a mathematical way, you do a Lie algebra computation in the usual way, compute Lie derivatives in the usual way, and you end up with the identifiability result. And there's something at the end about this. I'll skip that. I think that I'm going to probably skip that. Let me just say that. Yeah, I was talking a little bit more about the optimal control thing. Here's what I was telling about what the trajectories do. This is a typical trajectory. Actually, this is a cartoon, but it sort of gives you the idea of what happens. You get to you slide as long as you can, and then eventually you will get out, and that's when you lose the game. Since you were saying they're interested in optimal control, just set up the Hamiltonian, do the usual looking for abnormal extremals. You can actually show that, I think in the case of alpha equals 0, I had in one of those, there are no singular extremals in the usual sense of the switching function mean identity is 0 on an open interval. Outside of that, of course, we have band controls. This is basic usual theory, textbook theory from 70 years ago. But the part that is interesting is when you're on this, because of this problem being on a closed set, and the fact that you're talking about exiting the closed set, this things where you're moving along the boundary of the set as long as you can doesn't fit in any of the optimal control theory in the textbooks as far as we know. So we have to develop all that. So sort of cool that he develops a new mathematics in the middle of doing this, right? Which was find interesting. I think I'm going to skip. Yeah, this is all the calculations. And for those of you that know this stuff, it's the usual calculations with the switching function. You look at the right Lie brackets. And yeah, so that's, most people, I'm sure here are not terribly interested in that. But we have a lot of discussion in the second paper about the geometry of the problem. It becomes a pure math exercise, right? But it's sort of fun, right? But that's how you justify it. I mean, one of the things that you justify mathematically is that it seems that the larger alpha is, the more switching you have to make. And the question is, can you prove that mathematically? Is that the theorem that the larger alpha is, the more switching you have to do? And once again, it sort of follows from the Lie algebra structure of the problem. Let's see, was there, yeah. So yeah, I'm going to skip a lot of this. But yeah, like I said, you have to use under Clebsch at one point to rule out certain solutions. I'm going to skip all that. Yeah, identifiability, as I said, you do the usual thing, you compute Lie derivatives of the output, and you look at the space of observables, ask when that separates points. We had work that we did way back in the 1980s on identifiability that gives you the basic theoretical. Yeah, I think I'm going to skip all that. Is there something else in here? Yeah, so we also looked at things like sequential therapy, having two drugs. Should you apply them in sequence or at the same time? Only simulations. We didn't actually do anything theoretically with that. Yeah, so there's some things in that paper. And yeah, I don't think there was anything in there that was particularly, yeah, I don't want to spend your time with it. So anyway, so that's sort of the quick version of what I could remember from this paper. But the slides are very old, but I, where was it? Okay, summary of theory results, whatever. Anyway, so yeah, sorry. I thought I had like a, there was a conclusion slide somewhere. Is it the next one? Two more.
It's more.
More. Okay. Don't. Did I? Well,
It's after the identifiability.
oh, that was a conclusion. Oh, yeah. Okay. All right. So, yeah. So anyway, so to summarize then, the key point is the classical question that has been asked for the last, 75 years or whatever is resistance pre-existing, or does it arise during treatment? That's the classically in the case of bacteria and antibiotics. In the case of cancer and targeted therapies or chemotherapies, same questions arise. But the main point, if you don't remember anything else about this lecture, besides the pictures with the Bang controls and whatever, the main point is that we are insisting on the fact that when you talk about induced resistance that arises during therapy, there's a difference between resistance that arises randomly, whether genetically or epigenetically. Okay, the epigenetic ones will tend to reverse, the genetic ones obviously not. By and large, it's probability of 10 to the minus 9 of happening, you don't expect to happen twice. Okay, but the point is that there's a difference between pure selection of things that happen randomly or Lamarckian induction of variation, generation of variation through the treatment, and then Darwinian selection. So that sort of Lamarckian / Darwinian view is different from the pure Darwinian view. So we have three at least different mechanisms, not two. And I want to emphasize that because most people just look at the two. And once again, you could argue that it doesn't make any difference. And like I say, when I talk to oncologists, I talk to biologists, it doesn't make any difference. It's happening during treatment. But the point is that as illustrated by at least this very trivial mathematical model, it has tremendous potential implications for treatment. In one case, you want to do pulse treatment. In the other case, you might want to do continuous treatment. Is this a general message? No, of course not. I cannot say that. It's a message in a toy model. And of course, one needs to do much more to be able. Anyway, that's it.